The Double Helix, a personal account of the discovery of the structure of DNA, by Shannon Felice and Sammy Caputa. This book has a self-explanatory title, as the book is about the discovery of the structure of DNA. It was a scientific journal primarily written by James Watson in the 1950s that consisted of the battle to the discovery of DNA structure. This book demonstrates Watson and Crick's competition with rival Linus Pauling to the credit and fame of discovery of the double helix structure. Linus Pauling created the A-helix model for his first attempt at describing what DNA might look like. In November 1952, Watson discovered RNA coincided with DNA, and in December 1952, Pauling hypothesized a structure for DNA, which consisted of a sugar-phosphate backbone. In 1953, Watson and Crick discovered the double helix structure for DNA. While James Watson and Francis Crick are majorly credited with the introduction of the double helix, Rosalind Franklin was actually the mastermind behind it. Watson said she was his assistant, but in reality, she did her own research. Watson also slandered Franklin in an attempt to discredit her work because she was a woman in science. An individual scientist took Franklin's B photo from her crystallography picture of DNA structure and showed Watson against her will. And Franklin was also one of the first to use X-ray crystallography, which played a major role in her discovery of DNA structure. On the left side of the screen, we have a cartoon version of a double helix DNA model. It shows the hydrogen bonds, which consist of adenine, thymine, cytosine, and guanine. These play a role in gene expression, and thymine and adenine always pair together, and cytosine and guanine always go together. On the right side of the screen is a real picture of DNA through a microscope. If you look closely, you can slightly see the spiraling double helix structure with the hydrogen bonds in between. This illustrates how difficult this could have been to discover the structure, especially with a lack of technology in the 1950s.
DNA is an essential part of our being. Everything, even plants, have DNA. Through DNA, we have diversity of hair, eye, and skin colors, skin pigments, height, and more. Some even believe it gives you certain behavioral traits, which would be the nature versus nurture argument in a lot of scientific worlds. Through reproduction, we and our spawn inherit traits genetically through DNA and what is held in the DNA structure. It helps code adenine, thymine, guanine, and cytosine together in 2 protein sequences throughout the structure. Without it, cells would be unable to live. The structures play a key role in death because the double helix begins to unravel, leaving cells unable to reproduce. Without the discovery of the structure, we would not have the knowledge that we have today to understand concepts such as genetic mutation, cloning, diseases, especially those of genetic nature. And with the double helix discovery, it helped us engineer specific drugs to help specific diseases. Genetic expression, which sets us apart from other animals with the coding of DNA. Forensics, which with the help of the double helix structure led to the DNA database CODIS, which is used by many law enforcement agencies today in order to catch criminals, and in addition, helps forensic science in general. It can also help find what diseases you're predisposed to through the bloodline and who your parents are through DNA testing. Selective breeding is used to breed plants for durability, especially against pesticides, among other uses. Some people would like to use selective breeding in humans in order to choose what their children look like, act like, etc. Many agree that this would be unethical and dangerous for humans to do, and would mainly only benefit the rich in attempting to create a superhuman race almost. Any difficult science experiment breeds trial and error. In this research, many times they had to break from the research in order to finish it. Also in this case, they went through a few structures, including the A helix and the 3-chain sequence model, before discovering the double helix. Also, in this experiments, there was a few rivalries. Watson and Crick versus Pauling was the rivalry between battling to who could discover the structure first. For a long time, they were neck and neck. Also, Watson was trying to undermine Franklin's work, especially after she got sick and died, and he took most of the credit and she couldn't say anything. The technology of this time wasn't what was needed in order to help discover the structure. However, Franklin put good use to X-ray crystallography in order to get the first B photo to show the double helix structure. Multiple scientists were needed in order to discover the double helix structure. Although only Watson and Crick were credited, many know that Franklin should be and argue that Pauling should also have a say in the discovery.
We'd like to leave you with this quote that we liked from the book. Worrying about complications before ruling out the possibility that the answer was simple would have been damned foolishness. John Watson.
We hope that you learned something about DNA, although it might not be as much as we learned from this book. Please leave us any questions you may have. We look forward to answering them.